Hey, welcome back, ladies and gentlemen. Today, we are going to attempt to do a project on evolution through natural selection. And we are going to have a fictional population, and we're going to watch over how, over the course of 10 generations, this group is going to change. Something you need to remember, though, from the notes is that evolution is not about how animals change, how people change. It's not about, about how individuals do anything. It is about the entire population and how their genetics change, right? Not how the way they look changes, but how their DNA changes. And to talk about that, we are going to go back into last chapter's information. We're going to be using the words alleles. Remember alleles? The genes, right? If you have two dominant alleles, you're homozygous dominant. Two recessive alleles, you're homozygous recessive. A dominant and a recessive, you're considered heterozygous. You have a dominant and a recessive gene. If you have both, you're considered dominant still. We are going to see how that is going to change over the course of 10 generations. So let's jump into your handout, read through the directions, and talk about what kind of changes we are going to have to make because we're not doing this in the classroom. You're doing this at home, and you're going to have to make some adaptations on your own. All right, so let me screen share this. All right. This is called the Bean Bunny Evolution Project, modeling gene frequency change or evolution in a population of natural selection. Right. This is a weird term here, gene frequency. This is just a really fancy sciencey way of saying how often this gene shows up. Like if we're going to be talking about rabbits, bunnies, if you were to go out into the wild and count up all the rabbits in this population, how often, what percent of the time are these certain genes going to show up? Right? 10% of the time, 20% of the time, 30% of the time, because we don't say percent in frequency, right? We're going to keep them as decimals. So instead of 10%, it's just 0.1. Instead of 20%, it's 0.2. Got it? Cool. So let's go read these instructions here. In this activity, you will examine natural selection in a small population of wild rabbits. Evolution on a genetic level is a change in the frequency of alleles in a population over a period of time. Right? Change in frequency, change in how often you will come across those alleles. Breeders of rabbits have long been familiar with a variety of genetic traits that affect the survivability of rabbits in the wild, as well as in breeding populations. One such trait is the furless rabbit, or naked bunnies. This trait was first discovered in England by W.E. Castle in 1933. The furless rabbit is rarely found in the wild because cold English winters are a definite selective force against it. All right, I'm going to pause there for a second. Selective force. That means this is something about their environment that will kill them. Right? If you are a furless bunny, you will freeze to death in these cold English winters. So that's why we don't come across furless rabbits too often, is because they're dead. That's an important thing. That's called natural selection. If you're not good enough for your environment, you die. In this lab, the dominant allele for normal fur is going to be represented by a capital F, right? That's your dominant allele. And recessive allele for no fur is a lowercase f. Bunnies that inherited two big F alleles, or one big F and one little f, have fur. So big F or big F little f do have fur, they get to survive. While bunnies that inherit two little f alleles have no fur. So little f, furless bunny, they die. Sad. The purpose of this lab. In this activity, you will model evolution by natural selection of the gene frequency of two alleles in a two alleles in a population of organisms. All right, so we are measuring the change of those alleles in the population. That's important. You will also calculate the gene frequency of the alleles for each generation and then graph the frequency for two alleles over 10 generations. All right, so you go down here to procedure. It says working with a partner. You are not going to have a partner, all right? You're at home by yourself. That's going to make life a little bit harder for you. So I made life a little bit easier for you someplace else. Up here where it says calculate the gene frequency, I got that for you. All right, I set that up for you. And then graph the frequency. I set that up for you as well. All right, so we'll read through all this, but down here, this is your table where you would put the information and then the paper you do the graph on. I went ahead and made that for you on a Google Sheet so that you can type it in right there. You don't have to print anything out. So if you were to say get 12 rabbits, right? Go out in the wild, do a head count and get 12 rabbits. And then they are big F, big F. And you got 26 rabbits that are big F, little f. And I don't know, I'm making up a number here, 12 rabbits that are little f, right? They are dead. They do not survive. Right? What you are going to do, and this is the hardest part for you, okay? I took care of all the really hard parts. That's what all this stuff. This is all equations. You don't have to worry about yet. So this is not the hardest part you're going to have to do in terms of math. Figure out how many big F's there are and how many little f's there are. That's it. So let's find out how we do that. Say you counted out 12 rabbits that are big F, big F. Well, there are 2 big F's per rabbit. Each rabbit has 2 big F's. So that means 12 rabbits, that's 12 times 2. All right, if you had 1 rabbit, that 1 rabbit would have 2 F's. So 12 rabbits, that's 24 F's. And then over here, 26 living rabbits that have 1 big F each. We don't care about this little f. Cover that up, ignore that, do that one later. We just want the big F's. So that is 26 big F's. They only have 1 big F, so we don't multiply it. This one has 2 big F's, so we do multiply it by 2. So we got 12 rabbits with 2 big F's, that's 24 big F's, plus 26 big F's. 24 plus 26 is, that's 10, carry the 1, 50. All right, 50 big F alleles. And then how many little f's are there? All right, well, let's count how many little f's survived. Right, little f, dead. That's why I made this gray as a reminder that that's sad because they're dead. So we do not count the dead rabbits. We only count the living rabbits. So you have 26 rabbits that have 1 little f allele each. That's really easy. No math involved. Just move over the 26. All right. So in terms of surviving in our generation 1 rabbit, our first generation of rabbits, we have 50 big F alleles that survive and 26 little F alleles that survive. Nice and easy. Check this out. If you have a big F, you get to live, right? We don't ever lose the big F's. So you can go ahead and fill 50 in because we should never lose the big F alleles, ever. If you do, you did something wrong. Maybe you dropped it someplace, right, when you were counting. Right. So we only need to count them. And then look at this. This got filled in here. This was nothing. Now it is. It added your big F's plus your little f's to your total number of alleles. And then calculated your gene frequency. I did some math here for you. Here's the equation up here. What is the percent of the time it will come across big F? This would be 0.657, you can round up 0.66. That's 66% of the time you'll get a big F, and 34% of the time you'll get a little f. Add those together, you would get 100%. If they're percent, this is decimal, so add them together, you'll get 1. This is how you double check it, 1. If you did all this stuff right, that should stay a 1. If that's not a 1, you did something wrong. Now look down here. It auto-filled a graph for you. Right? From here on, I'm just going to pick some random numbers just to show you the graph change. 23, and the numbers fill in, 22, 24, 19, and you can see the graph is changing. Right. So I'm going to go ahead and erase all that because Those are just numbers that I made up. I didn't actually do the work. So if you're confused by that, go back, rewind, watch that again. That means you can ignore these 2 pages. All right, so what materials are you going to need in order to do this? It says a brown paper bag. I don't have a brown paper bag. So I'm assuming you don't either. Any bag, any container will do as long as it's not clear. All right, don't use any clear plastic bags. No glass dishes or anything. I'm using my lunch bag that I carry to work with me every day. 50 red beans and 50 white beans. I would have brought beans in the class with me. I'm not doing that. I don't have beans here. I'm actually going to cut up some paper and ball them up. So I have a piece of blue paper and a piece of orange paper. I'm going to cut strips. I'll show you how I'm going to do that in a second, and ball up the little strips. You can use whatever you can, just 2 different things. Maybe you have different color paper clips, blue paper clips, or red paper clips, white paper clips, whatever. Anything that you can get around the house. I'm not picky. They just represent something else. These are going to represent the alleles. All right, the red beans were supposed to be the dominant big Fs, and the white beans are supposed to be the recessive little Fs. Use whatever you got around the house. Marker and Petri dishes, again, I don't have. I'm going to use some plastic cups and write big Fs and little Fs on them. That's going to be containers. All right, so procedure. Working with a partner. Read the following problem question for today's lab. Again, not with a partner, by yourself, but go ahead and text people, text your friends, whatever, you're all in the same class. How does natural selection affect gene frequency over several generations? You are going to answer that. I'll probably have this on a Word doc. And at the end, you got these questions. I'll have them on the same Word doc. All right, concluding questions. So your hypothesis will be with them on a Word doc for you. You're going to answer that question. How does natural selection affect gene frequency over several generations? From your previous knowledge about natural selection, evolution, and genetics, answer that question on your student answer page in the space marked hypothesis. State your hypothesis in an if-then format, meaning if this happens, then that will happen. Include your predictions regarding how you think natural selection will affect the gene frequency in a population. State what you would predict about the frequency of big F alleles and little F alleles, or dominants and recessives, in the population of rabbits after 10 generations, where the little F bunnies are selected against and do not survive. Every time you get little F, they die, and you throw them away in a graveyard. Trash can, it's fine. The red beans, or whatever you're using, represent the big F allele for fur, and the white beans, or whatever you're using, represent the little F allele for no fur. The paper bag, or whatever you're using, represents the English countryside, where the rabbits randomly mate. Using the overhead marker pen, marker, whatever you got, label Petri dish. Again, I just wrote on a sticky note and put it on a plastic cup. And write big F on whatever container you're using for the homozygous dominant genotype. Label the second one big F, and label the third one little f. And that's going to be your dead rabbits. All right, now setup. Place 50 red beans or whatever, and 50 white beans or whatever you're using to be your alleles, in your bag and shake them up. That shaking represents mating. Please note that these frequencies have been chosen arbitrarily for this activity. All right, 50 and 50 just makes it a little bit easier. In the wild, who knows what we're starting with. Without looking at the beans, close your eyes. Select 2 at a time. All right, just take 2 beans or whatever out of your bag, and that is a rabbit. All right, 2 of them. 2 beans represent 1 rabbit because each rabbit has 2 alleles, one from mom, one from dad. That's what you're grabbing, all right? You're grabbing their alleles. Place that rabbit, the bean pair, into the appropriate dish. So if you get 2 dominant alleles, throw them in your big F container. Get a dominant and a recessive allele, big F little f container, and 2 recessive alleles, throw them in the big little f container. For example, if you draw out 1 white bean, 1 red bean, place both beans in your big F little f dish. Common sense. Continue drawing out pairs until all the beans have been placed in the appropriate dish. To determine the number of individual rabbits produced in each phenotype, count the total number of beans in the appropriate petri dish and divide this number by 2 since the individual represented by a pair of beans. Record your results in the data table. That's difficult to do. Just count them by 2s. All right. Just when you count them out, you are going to count out. Let me find down here. Number of big F bunnies. How many rabbits? So in your big F container, count how many rabbits are there. Not how many beans or whatever you're using. How many rabbits? 2 beans equal 1 rabbit. So you get 2 beans, that's 1. If you're getting 4 beans, that's 2. You want to count them out individually, that's over here. Why are we not counting them out individually? Because that's going to get a little confusing with over here as well, just doing what the paper says to do. This one wants to know how many rabbits there are. This one wants to know how many alleles there are. That's where you would count individually. Nice and easy. All right. The little f bunnies are born furless. The cold weather kills them before they reach the appropriate reproductive age. So they can't pass on their genes. You're going to place those beans from the little f container aside before beginning your next round. You will not count these beans in the number of little f alleles column. So you are going to count how many are there, keep record of how many died, but you do not count these for this column, right? You only count these. They're the ones who survived. These guys are going to go back and reproduce and make this next generation. These guys are dead. They do not reproduce. They do not get counted over here. Let's see. Number 8. Count the big F and little f alleles that were placed in each of the furred rabbit columns dishes in the first round and record the number in the data table labeled number of big F, number of little f alleles. Right, that is these 2 right here. So first count out how many bunnies there are, and then count out how many alleles survived. How many altogether? How many rabbits altogether? How many alleles survived? If you're confused by that, please get in contact with me. Repeat steps 6 through 9 to obtain generation 2 through 10. Make sure to take turns so that each partner has a chance. Okay, you don't have partners, moving on. Determine the gene frequency of F and little f. I did that for you. If there is time, your teacher may have you record your group's frequencies on the board. Not going to do that. Then complete the conclusion questions on the student answer page. These questions, all right? This is going to be on a Google Doc for you as soon as I make it. Graph the frequencies. That will be done for you right here. Right now it says no data. That's because we don't have any data in here. Once you put data in there, we saw that it filled up. And then we got some dots there, right? As you fill this in, the lines will move. If your teacher requests it, plot the class totals on the same graph. Okay, we're not going to do that because I don't know how to combine all of your graphs together, right? So all together, count out your alleles, put them into separate dishes. Once you do that, count out how many rabbits are big F as you go. I would keep a tally, might be easier for you. how many are big F, little f, and how many are little f, little f. These guys are dead. Bury them in the graveyard. And then number of big F alleles, number of little f alleles. Count out all of your dominant ones, count out all of your surviving recessive ones. And then take the ones that survived, put them back in your bag, shake it up again, and do generation 2 the exact same way. All right, and then you get 2 little ones, throw them out. And then the survivors go into generation 3. You do that for all of them. All right. If there are any questions, please feel free to get in contact with me. Again, as always, email works. Get in contact with me on Google Classroom works. And coming to talk to me during my office hours, normally between 9:30 and 12:30, Monday, Wednesday, and Friday. If you want to Zoom with me or talk to me any other time, email me and ask. We'll set something up. All right. Thank you. Have a good day.